Pleasure to meet everyone today. I'm Ernie Chilcott, based out of Durham, North Carolina, representing Hudson River Biotechnology. Currently sitting in central Netherlands outside, so forgive me if the wind picks up. I'll
Looks like a nice day.
yeah, it's absolutely gorgeous here. So,
Mm.
and of course, yeah, feel free. We'll have some time for questions at the end, but we'll just give you a little overview of the company and talk about a couple of platforms that we're working on developing at the moment. And let me get this into presentation. There we go. So Hudson River Biotechnology is an agricultural biotech company. We focus primarily on gene editing of a variety of different crops. We're based in the Netherlands, supported by public and private funding at this point. We're more than 55 employees. This has changed within the last week and will change again next week, so growing quite rapidly. And the company was founded in 2015, became fully operational from a lab perspective in 2018. Received a couple of awards over the years, and excuse me. Series A funding in 2021 was around 5.1 million. Currently working on around our Series B and another round, and the Series A funding was all about expansion, so double the lab space, double the office space in the current location. And the second round will be focused on developing our Pluntrance project, which I'll speak more of in a moment. Everybody's quite familiar with the progress that's been made in agriculture over the last 100 years. We've gone through phases of mechanization, agrochemical development, molecular breeding, GMOs. Two things that we see on the horizon is a need for greater sustainability. That was highlighted in the previous talk, quite well, I'll have to say. And then and maintaining yields. So we've made a lot of efforts, especially in the large agricultural growing regions such as the Americas and some parts of Asia, but not everybody is on an equal spectrum, if you will. So advances that have been made in certain regions don't necessarily carry over to others. And we think that we have a couple of platforms here that are able to bring everybody up to a level playing field. The first one is Mad Tiger. Mad Tiger is all about gene editing and plants. And the second one is Pluntrance. So, and that's about smart delivery of agrochemicals and biologicals. A little bit more about Mad Tiger. So CRISPR has been around for about 10 years. The beauty of this is in the precision. So you can basically focus at any given location within the genome. There are some caveats to that, but not necessary to detail in this presentation. You're able to disrupt a sequence by making a knockout or potentially influence gene expression in a regulatory region. You're able to insert sequences by making a knock-in, using a repair template, or influencing, again, a regulatory region within a promoter sequence. We're also able to do homology-directed repair. Influencing the expression can confer traits, of course, and that's the goal. And sometimes this is just one gene, or it could be a few genes, depending on the complexity of your target. And the diagram on the right details what it looks like with using an RNP for delivery and either deleting or inserting a gene sequence. The approach that Hudson River takes is transgene free, and it's designed to deliver homogeneous products in a single round of editing. So we start with many different explants, meaning the starting material of your plant can be done from in vitro tissue culture or leaf material. We isolate protoplasts, take them through incubation period with a RNP complex. And what that does is that's the machinery behind the editing. So that goes through the process of either making a knockout or a knock-in. And the right-hand diagram depicts how you go from a single cell, which is a protoplast without a cell wall, through a recovery of the cell wall and forming a microcallus. And then more compact calluses are going through propagation stages as we get to the upper right-hand side. And then we're introducing light and hormones in order to promote shoot elongation and root formation, and ultimately delivering a in vitro plantlet back to the end customer that they can use for propagation and or continued development in their breeding pipeline. So again, this is single cell transfection, transgene free, so no selectable markers. And depending on the regulatory region that you're in, this is considered a nude breeding technology. Some categorize that as a GM. Specifically, Europe is an outlier at this point, but many other regions throughout the globe have eased regulations and promote this technology. How is this done? So the big key here is all around transgene free, so no GM labeling. We have proprietary guide design softwares that are able to pick out sequences throughout the genome of your interest. We're using your elite germplasm and utilizing MAD7 nuclease for the CRISPR editing. We have seen on average editing efficiencies around 50%. Most recently, we reported more than 92%, or greater than 90%, sorry. Editing efficiency and agronomically important crop. We use single cell regeneration, so we're not producing any chimeras or any off-target effects with this. And we have homozygous plants delivered in single multiple rounds, editing of multiple genes in one single round. We have this working for more than 50, we're working on more than 50 varieties and 20 different plant species. I'm going to jump over and talk about our Plentrance technology, which is our all-around smart delivery of agrochemicals. Nanoparticles come in a variety of different shapes, sizes, and a variety of different uses. What we're most interested in are nanoparticles that penetrate cell walls, that are able to deliver a cargo, and we work with a variety of different cargoes. And these are biodegradable and that they have a good safety profile. And we talk about Plentrance technology has been developed for applications within the agrochemical space, as well as delivering unique fertilizer elements directly into plant cells. In addition, we're able to deliver nucleic acids and proteins. If you think about delivery of a naked molecule in many instances, a lot of times, for example, a fungicide is sprayed on the plant, it sits on the plant leaf. The lipid profile of the plant leaf maintains it on the outside, and it's never able to go systemically into the plant. Plentrance is capable of breaking down that barrier and going into cell walls. There are many different applications of this and needs for that. We won't get into those today, but happy to discuss them at a future opportunity. The most promising feature that we see with this is the ability to take a multitude of different cargoes and deliver them in an effective and precise manner. This technology was developed as an R&D tool to deal with recalcitrant crops, so delivering CRISPR to crops that aren't usually attainable to the traditional methodologies of delivery. And we are also actively developing this for larger scale applications and field applications. It's a quick example of delivering of a protein. In this case, it's neon green into a tobacco suspension culture. We are able to on the left-hand side, you have visible light. On the right-hand side, you have fluorescent light, and then a merge here. But we're able to actively deliver proteins. You can see fluorescence in the cell wall as well as in the nucleus in these two examples. That's a little bit about Hudson River Biotechnology. I wanted to keep it short today. Like I mentioned, we have our CSO, Ferdinand Los, and CEO, Rudy Aranis, are actively working for another round of financing and open to discussions with anyone at any time. Happy to take any questions that you may have.
Awesome. Well, thank you, Ernie. I really appreciate it. And I think one of the biggest questions I wanted to ask just out of the gate was I was impressed with how you guys approach the challenge of CRISPR editing and understanding that it it actually can be a lot more challenging to create great products or influence plants than just simply just editing the gene by itself. And that's why you guys built out that Tiger workflow, from my understanding. Can you give me an example of what are some of the challenges of when doing CRISPR editing for plants? What those challenges are and how you guys kind of overcame those?
Yeah, so our customer base consists of large multinational seed companies as well as the everyday small mom and pop seed company. We also work in the food and ingredient space and have a play in biopharmaceuticals, so using plants as a factory for proteins. And our methodology is was developed for making efficiencies and throughout the entire process. And there are a lot of steps as you go from a single cell without a plant wall all the way to a regenerated plant. And a lot of the times we have questions around certain aspects of the workflow. Regeneration is a known issue within plant tissue culture. It has been for a long time. So these are usually this is usually one of the reasons that we're approached. We have a lot of different techniques that we can employ that are not traditionally used within the plant biotechnology space, and this gives us a unique advantage in my mind. We're able to overcome those challenges as well as deliver on the intentions and have a flexible business model and ways of working with customers that are viewed as very beneficial from both sides. We're all about creating mutual success.
Cool. No, that's super helpful. And I know it's a pretty broad question too to ask. And you talked about the flexible just now, the flexible relationship you have with your customers. What does that actually look like? Are you guys, creating your own plant products and selling that directly to different types of customers? Or are you contracted by customers to identify a product and then you license it out? Like, what does that relationship look like?
Certainly, we have people that have a trait in mind that don't have a gene in mind, and we can work with them to help isolate candidates and move them through the entire workflow from end to end. We do develop commercial lines for customers. We also do research projects. So maybe a customer has a long list of traits that they want to validate genes for. We can do ways of working with that. Traditionally, our model is fee for service, but and we also work under a royalty model on top of that. We're we're not in the business of becoming a seed company. There are plenty of people out there, and that's a
Yeah.
difficult business. So we're there as the service provider to help provide them with a technological solution in order to advance their breeding pipeline.
Awesome. Well, thank you, Ernie.